Hello everyone, this is my AP Biology Chapter 17 review video on protein synthesis and mutations. This is by far one of the most important concepts in cellular biology. Proteins are literally what make up our entire body, and without proteins, almost no functionality could actually take place. Our entire immune system is based off of proteins, for example, and they are what help fight off any invasions of bacteria or viruses. So we're just going to go over like the miscellaneous concepts at the beginning. So things like the central dogma, the differences between eukaryotes and prokaryotes, and what a plasmid is. So the central dogma is pretty much just describing the general process by which a DNA molecule or a segment of a DNA gets converted into a protein. So the DNA first gets transcribed into RNA, and then the RNA gets translated into a protein through transcription and translation. Now to cover the differences between eukaryotes and prokaryotes. Well, eukaryotes have a nucleus and prokaryotes don't have a nucleus. They have what's called a nucleoid, which you don't need to know what a nucleoid really is. You just need to know they have it. But next up, eukaryotes have membrane-bound organelles, while prokaryotes do not. So some examples would include like the ribosome, where there's a plasma membrane in the nucleus, and the ribosome itself also has a membrane, so that materials can exit and enter through any method of transport that we've discussed in a previous unit. So then eukaryotic cells, they divide through mitosis, which we've already covered. So PMAS, prophase, metaphase, anaphase, telophase, while prokaryotic cells divide through binary fission, which we don't really need to know what that is. You just need to know that there's a difference in how the two types divide. And finally, what is a plasmid? Well, a plasmid is just a circular piece of DNA, usually found in prokaryotes, but rarely also found in eukaryotes. So the picture on the right just shows, what the central dogma actually entails. So in the nucleus, as you can see, the DNA gets transcribed into the RNA through transcription, and then that RNA leaves the nucleus. And then translation happens and the protein is formed. So there's three main parts to transcription. There's initiation, elongation, and termination. So initiation is really just how does the RNA polymerase know when to start the transcription, like on which specific nucleotide am I supposed to start on. So a eukaryotic promoter just usually contains what is called a top box, which is a sequence of adenine and thymine nucleotides. So transcription factors, which are just proteins really, they must bind to the DNA before RNA polymerase, the main enzyme involved in the entire transcription section, so that RNA polymerase 2 can bind in the correct position and orientation. And after those initial transcription factors bind to the DNA, there's going to be additional transcription factors that bind to the DNA along with RNA polymerase 2, which is going to form the entirety of the transcription initiation complex. So now, since this entire complex is formed, RNA polymerase 2 is going to start moving in the 5'3" direction along the template strand. So it copies the complement base pairs through the base pairs that are on the template strand. RNA polymerase then goes through elongation, so basically elongates or lengthens the initial RNA molecule through reading the complementary base pairs of what is found on the template strand. And that elongation is not going to stop until you get to the termination part, which is what I'm going to cover on the next slide. So just to summarize what initiation is really, because there's a lot of moving parts here. So you have a eukaryotic promoter, usually contains a Tata box. You have a repressor. Um, you have an activator. I think that's what it's called. I don't remember exactly. Don't flame me for that. That's not exactly related to this right now. But then the transcription factors are going to have to bind before the RNA polymerase II to indicate the position and the orientation of the RNA polymerase II once it does bind with some additional transcription factors. Elongation occurs along the 5' end. Sorry, RNA polymerase II moves in the 5' end along the template strand, elongates the RNA molecule through reading the complementary base pairs. Okay, then we have termination, which is quite complicated because we haven't actually covered this in class, but it's just in case you might need to know this, right? So at some point in the process of transcription, RNA polymerase II is going to transcribe a sequence on the DNA called polyadenylation sequence. And this sequence is going to be bound in place by proteins in the nucleus. So it's going to be held in place pretty much. So now once RNA polymerase II gets a few base pairs downstream from this polyadenylation sequence, the proteins that are holding it together are going to be released, which in turn releases that polyadenylation sequence and the pre-RNA molecule, which is going to pretty much end the transcription part of it and send it the released RNA transcript into RNA modification. So just to summarize, just to say that again, so it's more clear, RNA polymerase transcribes a polyadenylation sequence, which is initially balanced by proteins. Then once RNA polymerase gets far enough and it goes past that polyadenylation sequence, the proteins that hold that polyadenylation sequence in place are released. And those proteins are going to basically cut off that mRNA transcript or RNA transcript from the DNA, and it can actually move on to the next step. So next up, we have RNA modification, which is pretty much the process by which RNA is, modified. So initially, we have a 5' cap that is added, and this is really in place just to help prevent RNA degradation. So all of the base pairs that are on this 5' cap don't actually code for proteins, but just in case some sort of event happens where nucleotides are removed in a mass exodus for some reason, then the base pairs on the 5' end are not actually going to code for anything. So nothing is going to be harmed at all. And the formal name for this is just GTP cap. So a similar thing is going to happen on the 3' end. You have poly A tail added to the 3' end, which is really just a whole bunch of adenine nucleotides. And this is really just to prevent RNA degradation on the 3' end, same concept as the 5' GTP cap. So then you have the process in which introns are going to be removed, exons spliced together. So this is done through an enzyme called the spliceosome. This basically removes all the introns, so the non-coding regions, splices together the coding regions or exons. And then an important concept is something called alternative splicing. And this is how multiple genes can be encoded from the same RNA, because the sections on the RNA that are treated as exons can vary. So sometimes a section on that RNA is treated as an exon, sometimes it's treated as an intron. So because of that, there can be multiple versions of a gene that is expressed because multiple combinations, basically. So then we're going to get to, there's actually 3 different types of mRNA, so I'm just going to describe what each of those do. And then we're going to get an overview of translation before moving on to the details. So first you have messenger RNA or mRNA, which contains the complementary base pairs to the original DNA segment. So this is what codes for the actual amino acids through triplets of base pairs called codons. So someone like isoleucine, for example, which is just three nucleotides as a pair. So each codon is going to code for an amino acid. You can find that on the amino acid chart, which are the monomers to proteins. Then you have ribosomal RNA, rRNA, and this type of RNA basically helps build the ribosome and assists in begetting translation at the ribosome. Sorry about the small errors in the slides. There's like a few typos. That's maybe a bit confusing, but I'm narrating, so it shouldn't be too egregious. So then you have tRNA or transfer RNA, and this type of RNA is going to carry the corresponding amino acids to the ribosome and attaches to the mRNA, and it's going to know which mRNA codon to attach to because of an anticodon that's attached to the tRNA, which is just a complementary base pair sequence of what is found on the mRNA. And translation occurs in the nucleus, obviously, translation occurs in the ribosome. Again, that's another error, man. I'm sorry about that. I did this in a rush, apparently. So this diagram is basically showing the process of translation here. So blue structure in the background is the ribosome. The mRNAs is all the base pairs along this pink line where a mouse is. So each set of 3 is a codon right there. And then you have the tRNA, which is this yellow segment here. And then the base pairs where my mouse is, those are anticodons because they are complementary to the corresponding mRNA codons. And because of that, it knows that a specific amino acid has to go in a specific place because of the complementary base pairs. Now, the structure of the ribosome is a bit more complicated than you would have initially thought. So obviously it has a large and a small subunit. But within the large unit, there's going to be 3 chambers. There's the A chamber, so that's the amino acyl tRNA binding site, the P site, which is the peptidyl tRNA binding site, and the E exit site chamber. So the tRNA is going to initially attach to the complementary mRNA sequence at the A site. And then it's going to move to the P site. From the P site, there's going to be an opening in which the amino acid is actually deposited. And then the tRNA is going to exit through the E site. And then, yeah, like I said, the protein is going to get formed through an opening in the P site. And mRNA, as I pointed out in an image in the last slide, is going to be basically along the intersection of this small and large subunit. You can see the A site, the P site, where the actual protein is formed, and the E site right here, where the tRNA exits and then maybe returns later for an inner amino acid. So mutations. Mutations are basically unintentional changes to DNA usually caused by errors in replication or protein synthesis, and sometimes like the effect is going to be very varied. Sometimes mutations are good, sometimes they're neutral, sometimes they're pretty bad. Really just depends on what amino acids are changed and what the resulting protein is going to do. So point mutations, they're basically just changes to one amino acid. So maybe a substitution for an adenine to a thymine. Then you have a frame shift mutation, which are deletions or insertions that are going to alter the reading frame of 3 nucleotide pairs. That's going to significantly alter the entire protein, because if you start at the beginning and there's an insertion there, each pair of 3 is going to be different from what it was before. So the protein is going to be composed of entirely different amino acids. And you have, so now these are not different types of mutations, these are more categories of mutations. So first you have a silent mutation. So these are mutations that don't change the amino acid sequence. They do change the actual nucleotide, but multiple codons can code for the same amino acid. So that's what's happening in this case. Then you have a missense mutation, which leads to one amino acid substitution. So an adenine to a thymine, maybe. So this results in maybe minor change to the function of a protein, but if it's an important sequence, like it's controlling the entire flow of what's happening in that particular area or organ or structure, wherever this protein is going to go, that one protein could have a major impact. It could, but it doesn't have to have that major impact. Then you have nonsense mutations, which lead to significant changes to the functionality of the protein because you're causing a change in the reading frame. So I already discussed that. And that will conclude this review video on chapter 17. I think this was pretty informative, except for a few errors in the slides, but what I said for the most part was correct information. So just listen to what I'm saying and you will be able to understand this unit pretty well. So thanks for watching, and I'll see you in the methylation and gene expression unit that's coming up next.